Hi, Ziyang. It's great to have you with us today. Thank you very much for being with us. Um, if you don't mind, I would just start with reading a little bit of your resume, so we are sure we are good with what we know about you, right?
Of
And
course, of
it's
course.
your opportunity to correct my mistakes, and there are some, unfortunately, very often. So you grew up in China, in Taizhou, right, and attended Peking University, where you majored in chemistry and computer science because just one was too easy, right? So you then traveled to the US and for your PhD to nonetheless Harvard in the group of Andrew Myers. And then you move on to University of California, San Francisco for your postdoc um, with the Demeronian Cancer Research Foundation. And you are supposed, and maybe you have already started to start your own lab at Berkeley. I don't know. That's a good question to start from. Have you started that, or are you still doing your postdoc?
You got both parts right. I'm still doing my postdoc, but I'm secretly ramping up my efforts to start my lab.
Okay, that's good. When are you planning to start? Do you have an official date yet?
Yeah, the official date is gonna be July the 1st this
Okay,
year. But I have started talking to the prospective students, trying to lure them to Berkeley and perhaps do a little bit of research with
of
me.
course that makes a lot of sense. But you keep being busy at San Francisco, still full on your current project then.
Yeah, yeah, still never put down science.
Okay, of course, science can be put down. All right, so the usual way we do this is starting from having a look backwards. Okay, so um, one of the things that we really like of these formats is trying to put a link between the science and the person. It's really fascinating because you end up seeing, right, the person through the science, and the best way to do this is just, you know, reconstructing your path. So I just want to know who is Ziyang, right? So let's start from traveling back in time and just reconstruct slowly your personal and professional life. And uh, you know, uh, it's obvious to start from where did you, how did you stumble across chemistry, science and chemistry in particular? When was that?
Yeah, yeah, no, I like this type of exercise because sometimes I even forget myself. Uh, I'm Ziyang. I always think of myself as a student of science, as student of chemistry. But looking back in time, it's hard for me to make a clear-cut uh, line where the point was that I started to become interested in chemistry. When people ask this type of questions, I often get nervous because there's some expectation that I have a fantastic life story that just turned me into chemistry and I'm not looking back, etc. But the reality is not like that. I think, um, but if I do have to rewind my life and look back, I feel like since a kid I'm always on the nerd side, if you will. It's like I like uh, reading about these weird facts in science and thinking about how the universe runs. But these are all general uh, science imaginations, if you will. I do think I have a interesting family setting because my mom works in a hospital. So I think by influence I get to hear some medical terms sometimes, and I, for whatever reason, like to read the packet inserts of the medicine I'm taking. Uh, it's just as a kid, maybe that's what got me started. Um, but yeah, I really liked the feeling of not knowing something and not knowing how something works, and then getting to figure out how it works. Either it's by reading or by doing the experiments myself. I feel like if I had to start back when I was a kid and take a 1,000-foot view of how I progress. This has been how I liked things to be
Were
done.
you that kind of kids who just disassemble things to see how they work?
I did a few. I yeah, I only did the disassemble part,
Yeah, of
but
course, you never reassemble.
that's one thing I
Yeah,
really
yeah.
liked.
Yes,
Yeah.
of course. You know, you sounded kind of worried about the fact that you didn't have this sort of bang moment, or this kind of grand story about how you got into science. But most people are like that, which is what makes it fascinating. And in many cases, you even just end up in science by chance. It just happens. It might be the person you meet. It might be that you're just good at school in science or math or whatever, and then the opportunities come along, and you embrace it, right? And so it's actually quite interesting how humble the origins of some great scientists can be, and it is really interesting to me. you know it makes it interesting. But you were a talented student at school, right? You went to chemistry Olympics, and you seem to have had a quite strong resume, even as a young kid.
Yeah, I did do all of those, and I also did things that aren't on my resume, um, such as doing crazy reactions with my high school chemistry teacher and burning the floor of the classroom. So these are things
Now I
you
want
wouldn't
to
learn
now I
about.
want to hear more about that. What did you do?
It's I'm struggling with the English term of it. It's when you mix aluminum and iron
Okay.
oxide, and then you heat the whole thing up, and then there's always lots of fume, and then super hot iron drop onto the floor. Um, yeah, that type of thing.
Yeah. Well, I guess you get to know that chemistry can be dangerous from early on, right? So then you pay attention as you grow up and you do more serious stuff, isn't it?
That is very true. Yeah. I would say the more I venture into chemistry, the more cautious or prudent I've become, just knowing how powerful it can
Right.
be.
How did you get the opportunity to go to the US for your PhD? That's quite a thing, isn't it?
It is a thing, but honestly, when I was in college, um, there's all sorts of directions my classmates went. Some of them went to Europe, some of them came to the US, some of them stayed in China. Some of them just ended up flying around the world because they decided they were going to do business instead. Um, for me, it was what I wanted to do because I did think that I wanted to do some organic synthesis for a PhD, for chemistry PhD, and um, the best organic synthesis labs uh, were either in Europe or in the US, and I felt like I really liked the way my PhD advisor Andy Myers addressed these problems. So it was really the science that attracted me to the US, not the other way around.
It is interesting, and it is a fact that science has this sort of multicultural, international environment attached to it, right? And it kind of makes it easy in some ways. I still feel I'm an expert myself. I still feel like it's a major move, especially when you're still relatively young after high school or after uni. Just going abroad where they speak a different language, and there's a completely different culture very often, and there's it's not a straightforward thing. It needs skills, right? And probably some sort of courage, strength. How was your experience? Was it difficult?
It was. I would say at the beginning, of course, it's difficult. There's culture shocks. There's language barrier. It's after all a completely different country. Um, but I would say one of the things I like to do is to try things that I'm not good at, or I just have completely no ideas about, and coming to a different country is one of them. So you see, it's the fact that something's unknown to me. I don't like that. I want to put some effort in trying these new things. Not crazy, reckless things, but new things that could be interesting.
Feels like a great skill to have for a scientist, right? And do you think that you're sort of pushing yourself towards the unknown and facing these things, and your curiosity gives you a bit of an edge? This extra experience, is this something that makes you different as a scientist, in your opinion?
Hmm, that is a hard question. I I get asked that a lot, and every time I have trouble answering it. But this time, I think I'm gonna say something different because when people talk about a international scholar, international student moving from one country to another, they tend to ask what's so different. But I feel like more is in common, and people don't talk about this very often. I just feel like the science always is the common language for us. When I moved from China to the US, I never had any trouble with doing the science because the science is the same. We speak the same scientific language. And honestly, there's a lot of personality match among scientists, despite they're from different areas, parts of the world. I find this fascinating because it it is a field where you can just have people from all places over the earth come together and they have something to talk about. I feel like that's really unique for science, and that really helped me along my way. And I think in terms of um, cross-cultural scientists or international scholars, uh, that's something we should really, really appreciate. And that, let's just not forget the reason why we can come together is that we are all excited about the science.
As I was looking into your work with interest, I found it quite difficult to define it. And I know it's bad to put labels on things. You shouldn't do it really. But you are an organic chemist by heart, right? You mentioned synthetic chemistry. That is where you're coming from. Most of your work is on drug discovery related in a way or another, right? But unlike most people active in drug discovery or medicinal chemistry, you are very diverse. People tend to specialize on specific targets. You have done work in multiple different areas, right? It's unusual, I would say, right? I'd love to hear your perspective, and if this is just happened, is it because of the labs you ended up working in, or is it because of your own curiosity and approach to it?
I would say it's mostly from curiosity, and
Hmm.
if you there's some sort of evolution in terms of my own uh, thinking here. When I first started doing science, I chose to do chemistry. Well, I was confused at the beginning. I wanted to do computer science, but the moment I chose to do chemistry, I was it was because I liked creating things that didn't exist in nature, right? That's organic synthesis, making new molecules, making complex
Yeah.
new molecules. That's how I started, and I just wanted to make the fancy looking molecules. But then, uh, during grad school, actually at the beginning of grad school, I realized, well, these molecules have to be made for a purpose, and that's
Yeah.
why I got interested in Andy's lab and joined his lab and started working on these antibiotic molecules. They It's a transition for me because I was still making very complex natural products, but now I have a purpose. I want to solve the problem or partially solve the problem of antibiotic resistance, and that's what drove me. Uh, my that's my drive. That was my drive to keep cranking it, because honestly, making these complex molecules wasn't that easy, and you'll
No,
see
of course
it
not.
took years of work. Of course, it took years of work to figure out how to make a single one, and then we figured out how to make a second. After that, it was easier. But that is that was the transition for me when I realized, okay, now I'm actually making something that's solving a health related problem, a disease related problem. So I wanted to do more of it. But who am I? What am I good at? I'm good at doing chemistry. I'm good at making molecules. So this is going to be my approach. I would like just to use what I know about chemistry and and do tricks, do chemistry tricks on the problems that aren't solved yet. And that was partially why I came to UCSF and why I got attracted by Kayvon's research, because that's exactly what ISLab wanted to do. It's never about making a single class of molecules. It's never about doing a single type of reaction. It's never about working on a single protein. It's figuring out where the chemistry problem is and find a chemistry key to that lock.
And it is really fascinating because, well, I can see the person behind it, right? You mentioned since the beginning of our chat how you like to push the envelope, get out of your comfort zone, you know, uh, understand the unknown, right? And here's your curiosity, here are your choices, and going in a lab where there's potential, a lot of potential directions where you can move into, and and what is interesting now is you saying you obviously like the challenge, and you know you're a chemist at heart. You want to do molecules, but you need a purpose as well at the same time. So you start from the fundamental curiosity, understanding the unknown and the intellectual challenge, if you wish, and now you add the purpose, because that makes it obviously better, right? So as you mature and you carry on, what is becoming the most relevant to you? What takes you out of bed at night? Is it still the thrill of the challenge, or is it more the fact that your work can really be meaningful for others?
It's both. It's both. I think nowadays I would say it is it is the potential impact, it is the potential consequences of what I do that gives me the fuel. But then every day, what gets me out of bed is really questions like, oh, what if I just made this molecule? What if I tested the molecule on this mutant protein? You see these problems become more granular on a daily basis, but you see how the final push is again, I want to solve this problem, but then I have to think chemically. Then I ask chemical questions. Uh, that's what keeps me going, and I feel always excited about that because, as you said, there are two ways of, of satisfaction or fulfillment. One is to make something impactful, the other is to venture into the unknown, and I can do both. And that's probably why I still very excited about what I do.
It is really interesting. So uh, there's a lot in your work that can satisfy both things, right? A lot of challenging projects, a lot of interesting applications. So you started from your work on the microlides and the antibiotics. Then you're working on cancer, the alcohol abuse dependency, and even chemical immunology, even COVID-19. And by the way, you haven't started your independent lab yet, and you already have a Nature, a Science, and several other high impact factor journals, right? So articles. So it's just it's quite amazing, and I genuinely don't know where to start from. Shall we start from the antibiotics? Because this seems to be like a sort of following a chronological order. You started from making complex molecules that also have some applications, and well, the idea of fighting antibiotic resistance is quite a subject, right? Do you want to start from there? Because there's a lot of interesting science there. You're trying to make these natural compounds in the lab is feels quite a task. By the way, I would like to start from the why. Why even venturing there?
Yeah, no, I started grad school at a lucky time, I think, because it was when ribosome crystallography was taking off. The Nobel Prize was just awarded to three scientists because of their work on solving the ribosome structure. And that was a good time for me because these antibiotics, the way they work, especially the macrolide antibiotics, was that they hit the bacterial ribosome and prevent the bacteria from making proteins that they rely on. And so scientifically, the pieces were coming together. There's structures telling us how we can make new antibiotics, and at the same time, there's also a what's known as the antibiotic crisis in 2007. That was the lowest moment in the history of antibiotic discovery because it's traditionally not a very profitable field. Not many companies are investing in new antibiotic discovery. So you see, the challenge was there, which is no new antibiotics, but there are new superbugs that are threatening the health of people. There is new science that allow us to see how things work, and then I joined a total synthesis lab, so there's this tool for us to make new molecules, and so that's how I got really fortunate to start my PhD project at that moment. So it becomes a natural choice for me because I want to solve this problem. And this lab really has great experience in antibiotic discovery. And the macrolides are a class of very complex molecules. It checks all the boxes. So I decided to get on that project.
Indeed, and very often, not usually, but very often, these total synthesis types of projects are more a sort of stretch of chemical synthesis. They are a motivation to look for different synthetic approaches, or a challenge per se, but they rarely end up into proper applications. You don't really make natural, particularly the more complex natural compounds via 27-step synthesis, typically, right? So using biotechnology methods remain the go-to method for those. Do you see this landscape changing, particularly for this class of compounds where you have worked on? Because at the end of the day, you develop an interesting platform there that allows you to create diversity. So what is your take?
Yeah, I'm glad you mentioned that because that is seemingly a widespread misconception about total synthesis. Because it sounds like a lot of work and you don't make a lot of the final product. So
yeah,
naturally you would think it's not that practical. But I want to argue the opposite because I think it is the way to make things that are difficult to make, but otherwise are the perfect drug. And in doing this project, we did want to send a message that it is possible to make large amounts of antibiotics, and you can essentially make any changes to the molecule as you want. Traditionally, these macrolide antibiotics are made by fermentation of cultures and then extracting the product out. That says you can only make what nature is good at making. You can do a little bit of chemical decorations on the stuff you extract from the fermentation process, but that's very limited. It is preferred only because it's convenient, and I'm not saying convenient is bad, but convenient means more economical, less time and money investment. But the message we wanted to send out is if you are willing to take on the risk and work hard on this type of problems, you can make a very practical synthesis. And that was the guiding principle of our work. We wanted the synthesis to be scalable. We want to be making grams or even kilograms of material. And we kind of wanted to change the widespread conception that total synthesis is just not related to reality. It's some toy that organic chemists play. that's simply not true. If
It,
you have the dedication to build that bridge.
and it's really good you're going there. You're anticipating my next question, because I would have asked you to summarize the results of your work, right? But you're saying, one of the biggest novelties was that you really aimed at making consistent amounts, to demonstrate that you can make useful amounts of these materials. So your platform really is thought for practical use somehow. So, but I think it's much better if you summarize the importance and the results of your work rather than me trying to do it.
Yeah, I think, well, I kind of just answered that in what I said earlier. The importance, I think, what makes me excited about this project, I should say, is first of all, it really addresses a problem, because bacteria, they learn how to evolve. They really outsmart us. They can evolve every 30 minutes, but we humans can't. So what we can do is use our science to win. And another one is, again, I want to show the society that chemistry is really useful, and
Sure.
it's just that if you get at it, and if you have the goal in your mind to make something that can be practically adopted, you can do it. I want to say there are approved drugs that are made by total synthesis. Hydrochondrin is one good example. It's an anti-cancer drug. It is made through a 30-ish step synthesis, but it is approved drug. It works on patients and really helps numerous patients fight their cancer.
Indeed, and that's a good segue, right? So you mentioned cancer. Some of your more recent work has been on that. I found really fascinating your work on the glioblastoma with targeting the mTOR. I'd love you to describe a little bit of that work, and it's really fascinating to me, because from the execution and scientific perspective is truly different from your work on the antibiotics, right? So from the macrolides, the challenge was really the synthesis, and then the application was kind of a plus. So you were doing it with a purpose. Here, it seems like the pharmaceutical target and the strategic approach is the challenge. It's a completely different scientific scenario. I was really fascinated and kind of surprised with these two so different types of work coming from the same person.
Yeah, well, it's interesting you said it that way, because I tend to think about this differently. It's the theme has been the same for me. The question out there is always, here's a disease, and the disease hits a human body, and then you want to find a way to attack the disease, but not anything else. Now,
Right.
for bacterial infection, that's easy, because the bug is the bacteria, so naturally you want to do something that only hits the bacteria. And then you have many options. You can hit any component of the bacteria. In fact, that's how most antibiotics work. And I chose to work on the macrolides because that's a very nice way to hit the bacterial ribosome without hitting the human one. Now, it's the same logic in the later projects too. Now, if you think about glioblastoma, it is a cancer that originates from a patient's brain tissue. And the goal is to hit the cancer, but not the rest of the patient's tissues. So how do we do that? We don't have a different species now, but a glioblastoma is a brain tumor. It is most of the time localized in the brain. So what if we could concentrate our drug to the brain tissue without affecting anything else? So it's still the same problem. How do you use a chemical trick to only attack the disease? But now if you follow this, you have to design a different approach, because now if we're working on human tissues, or a disease that originates from human tissues, a lot of the things are going to remain the same. So by focusing the attack on the certain tissue type, in this case the brain tissues, you can, concentrate your ammunition, if you will, but also avoid hitting the healthy tissues. And plus and a minus, that's the selectivity factor there.
Yeah, it's interesting because you're putting your chemical lens through to the problem, right? And for you, the perspective is the same. But I find it interesting that you almost disregard, how different the type of problem and the type of
No, I'm not
scientific approach.
trying to evade the question. I think it is a different project. From the beginning to the end, the ideas are different. But I do really want to focus on the common things because that's how you can continue doing solving different problems using the same philosophy.
Okay, let's describe the project a bit more in depth. Can you tell us about describe what the problem was and maybe doing a little bit of background on what the target is and how it works and then we kind of try and make sense out of what you said. You're trying to focus your ammunition in a specific tissues against another. So we're gonna understand how you did.
Yeah, of course. I'm happy to. Back to glioblastoma, it is an aggressive brain cancer, and we don't have many ways to treat it, depending whether you're asking a physician or asking a chemist. You get answers like chemotherapy, radiotherapy. Surgery is difficult in the brain, but it's also an option. But other than that, we're pretty much out of ways to treat patients with this devastating disease. And then here comes the important part, because these glioblastoma cancer cells, turns out they activate a particular pathway so that they can grow in an unlimited fashion. And this pathway is called the mTOR signaling pathway. And it's a protein I'm sure you've heard of. It is kind of the central regulator of the cell. It senses how much nutrient is out there. It senses how much stress is out there, and it tells the cell how much to grow and when to grow. It's a protein kinase, so it sends these type of instructions in terms of protein phosphorylation. Now, what these cancer cells learn to do is just to have a very active mTOR. So, it doesn't care if it's a very stressful situation or there's not much food around. It just wants the cells to grow. So, okay, now it becomes clear, if you can shut down this mTOR in the cancer cells, they should stop growing. And that is exactly true. You can shut down mTOR in these cancer cells and they will stop growing. In fact, in some cases, the tumors will shrink for a bit. There's only one problem. If you shut down mTOR, how do you only shut it down in the cancer cells? If you shut down mTOR everywhere, you basically shut down the human body. Now, it's not as bad, it's not as severe as I put it that way, but the truth is, if you use a mTOR inhibitor in patients, there are all sorts of toxic side effects. Patients have immune suppression, they get metabolic problems, and it becomes dose limiting because you simply can't use that much mTOR inhibitor in a patient.
So mTOR is not something that only exists in the cancer cells. It exists in all the cells of the body, right?
Exactly. So then here comes the necessity of only inhibiting mTOR in the cancer tissue. And then our approach is to think about it as a tissue-specific problem, to only inhibit mTOR in the brain. Now, when we convert the problem that way, we can reiterate the question in a chemistry way. How do you find a compound that only goes into the brain? Now, that's very hard. People have worked on this problem for years. They have used antibodies to navigate a compound, or to have a compound only gets turned on in the brain. But our approach is slightly different. What we thought we can do is, instead of navigating this compound into the brain, what if we could squeeze it into the brain? We tell it where not to go. We block all the roads except the road to the brain. So that's the premise for our chemistry design. And now, in terms of chemistry details, how this works is that the mTOR inhibitor we use is a rapamycin analog. So this type of molecules, they're quite special because they don't work by themselves. They need friends. They need a friend called FKBP. And it's a different protein, but the way these mTOR inhibitors work is they borrow the help from FKBP and stick itself onto mTOR. So now mTOR has not only a compound but also a protein on it. And now it's this steric effect that prevents mTOR from doing its work. So now, knowing this, here comes the chemical trick. What if we could steal or block all the FKBP that's not in the brain? Now, if you do that, you're essentially putting roadblocks on every single other road other than the one leading to mTOR. So that's what we did. We found a way to block the FKBP outside the brain. And then as a result, you're squeezing our mTOR inhibitor into the brain. So you're dialing down the systemic toxicity, but also concentrating our therapeutic effect in the brain tissue. So that's how I would put it.
So basically the idea is sort of two drug therapy, right? With one that goes into the brain and does the work as you want, to inhibit the mTOR, and another one which actually doesn't do it, or inhibit the effect of your rapamycin analog that actually can't get into the brain, right?
Exactly.
So this is what you call the rapa block, isn't it?
Yes, exactly. So it's a combination, as you said, two drugs, rapa link 1 and rapa block.
So how did you make it tissue selective? Is it just you made it more polar so that it can't pass the brain barrier?
Exactly. It is going against what people usually want to do. Normally, you want a thing that gets into the brain, but we do the opposite.
Usually, the drug design is kind of making a molecule that passes the brain barrier, right? In your case, you actually wanted to do the opposite, which is kind of interesting.
Yeah,
but chemically, doesn't matter, does it?
It's easier. You do everything that has failed.
Yeah, and how well does this work?
It does work really well. We have gone so far. We have tested this approach in mice with tumors in their brain, and we can stop the tumors from growing, but we don't get the liver toxicity or the immune suppression we typically see with systemic mTOR inhibition.
Yeah, that's really nice. Do you think there is a future for this? Do you see these studies progressing and maybe even getting into clinical studies at some point?
I'm looking forward to that. We haven't right now. I don't think there's direct clinical investigations on our approach yet, but I do think it has to do first efforts in time, and then finding the right disease application. Glioblastoma is one, but there are also many other diseases that's caused by overactive mTOR in the brain, and those could be interesting applications as well. We'll have to just address them in a case-by-case basis.
So is one of these the alcohol abuse disorder?
It is indeed. It is.
If I understand correctly, it's kind of similar problem. So, if I understand it correctly, it's kind of similar problem, similar protein. Is it actually? It looks remarkably similar to me. Am I right?
Yeah. Chemically, it is the same problem. I do think it is. I want to give a big shout out to our collaborator Dorit Rong's lab, and they figured out that drinking alcohol, especially in patients with abuse disorder, um, they get overactive brain. They get overactive mTOR in their brain. So, so that was where the original idea is from, because if that's a reason why uh, this this rewarding process, patients who have the problem would like to drink more alcohol. If mTOR is a culprit of this vicious cycle, maybe inhibiting mTOR is the right approach. And then it was um, it was a very casual meeting between our lab, between Kayvon's lab and Dorit's lab, that we decided, oh wait, you have a way to inhibit mTOR in the brain, and I want to inhibit mTOR in the brain to solve this alcohol use disorder. We should just collaborate, and that was the most smooth collaboration, I would say, and the ideal type of collaboration I, as a scientist, imagine.
It's great, and I can see how your chemical way to look at the problem makes it fun and fascinating. It's really coming through, Ziyang, your personality and your way of looking at problems and at life in general, and I can see how this translates in your type of work and in the diversity you have there. So some brilliant approaches. I'm really impressed. So and you've actually done more, and if this podcast could last 2 hours and a half, we could probably go on describe a lot of your other pieces of work. But I'm sure you have better things to do than spending your whole day with me here. So we are going and approaching the sort of final part of our interviews. But before getting there, I'm just curious, Roy. It's really difficult to predict now for me when you will be going next. So what are you planning to do at Berkeley, if you can tell me?
Yeah. I first thing I'll continue to do chemistry. I do think,
Of course.
I have always had this interest in our immune system, how it works, and how it failed to work. There are diseases where you want the immune system to attack cancer, for example. There are settings where the immune system is attacking ourselves in an unwanted way. For example, in type 1 diabetes, that's how the beta cells get destroyed by our own T cells. So, but it seems that we don't, at the molecular level, we still don't fully understand why these happen, and can we tweak it in a certain way? Can we make our immune system work harder, or can we tell it to hold on? This is friend but not enemy. We should stop.
Right.
That's something I really want to do, and I think being a chemist, again, I want to use the chemical approach, and that will be a main focus in my lab at Berkeley, is to use chemistry to control how T cells work or how hard they work.
Sounds really exciting, and I'll be sure to keep an eye out and see what you come up with. I'm really looking forward to that. This has been a brilliant conversation so far, Ziyang. It's always impressive to do our interviews, because I'm privileged to speak to so many smart people doing incredible things, and you're definitely one of those. And you're still very young. You haven't even finished your postdoc yet, and you have already achieved a lot. You're indeed an extremely talented chemist, and I'm sure there's a lot of great things that you will do down the line. But still, being so young, I think you still have done enough to be able to stop for a moment and look back and kind of think about what you would suggest to a younger scientist, or maybe a student, someone just starting in their career. What would that be your hint?
I could talk about this for many hours, but I would say the most important thing for me is to always keep a positive attitude for everything, for the research problems, or for things you encounter every day. I think um, here was this is what kept me going. There's always one new thing to try. You should never stop trying new things. There are many ways to put the same thing I just said, but I would say there are 100 ways to feel negative about what's going on right now, but you really want to aim for the positive one because that is a powerful one and that's an interesting one. And it's also the challenging thing to do when things aren't going well, but you want to stay on the positive side. And that's what's so exciting. That's what you want to do. You want to do the challenging things. So, so yeah, always keep a positive attitude is what I would say.
This is a fantastic message, Ziyang. Thank you very much. It's been great. It's been a great chat. I had a lot of fun, and I learned a lot. And I really applaud your attitude and your positivity, and the smile you have on while you're speaking about what you do. It really tells a long story. Congratulations again, and really thanks again for being with us.
Thank you so much. Pleasure is mine.